All right, part 4 of this lecture is to discuss differences between DNA and RNA. So some of the key differences, hopefully you're already aware of. DNA bases are A, C, T, and G. So one of the key distinctions is the use of T as one of the bases. DNA is usually double-stranded, so it's almost always found in its double-stranded format, and it contains a deoxyribose sugar. RNA bases are A, C, U, or G, so the T is replaced by U. So if you see a uracil used as the nucleobase, then you should know that this is an RNA. RNA is usually found as a single-stranded molecule, so either an mRNA or a ribosomal RNA or a tRNA, or even a small RNA. Almost always only one strand of RNA is found. And that's just the intent of the cell for what it's used. So because it's not carrying the heredity information, it's actually used more as an enzyme in these situations. And so it actually has some sort of physical action. It contains ribose sugar, and then it's also a much less stable molecule. So it's much easier to degrade RNA than DNA in a sample. So how do you recognize RNA? Almost always you just look at the 2' sugar. So if you look at the 1', 2', 3', 4', 5', the 2' carbon can either be bound to a hydroxyl group, an OH, or it can be bound to an H, a single hydrogen. So if you only see a single hydrogen, it's DNA. If you see an OH, then you know that it's RNA. So this addition of an oxygen, this hydroxyl group, can also actually attack the phosphodiester bond, and that's why RNA is a much less stable molecule. So here we showed you 2 nucleotides of DNA that are bound together versus 2 nucleotides of RNA that are bound together. And so you see that this extra oxygen group is there, and this oxygen group is actually very close to this bond. And so oxygen is a very reactive molecule in a chemical sense, and so this oxygen can actually form a chemical bond. And so if this oxygen attacks this bond, then what you do is you cleave the RNA molecule at this position. And so this is one of the important reasons why DNA is much more stable than RNA, and probably why it's used for the storage of genetic information. By losing this oxygen molecule, you've created something that you don't have to worry so much about it degrading just through natural chemical processes. So there are 3 main classes of cellular RNAs that are used throughout the kingdom of life. So messenger RNA. So messenger RNA are the templates. They transcribe a gene, and they're used to produce a protein. So the ribosomes use messenger RNAs to produce proteins. RNA molecules are actually part of the ribosomal particles themselves. So ribosomes are composed of a large number of subunits. Just in part, some of them are RNA molecules. So typically, there are at least 3 RNA molecules within the ribosome, and these class are called rRNA. So if you see the little r in front of it, that means that it's an RNA that's part of the ribosome. And then finally, transfer RNAs, tRNA. So these are also used for translation. They are not part of the ribosome, but they're used by the ribosome in order to attach amino acids to polypeptide chains. So we'll cover all of this in greater detail, but just you should know the 3 categories of RNAs and kind of how they're used in the cell. So they originate as complementary copies of 1 or 2 DNA strands during transcription. So DNA is transcribed in order to produce these molecules. So I've already kind of covered what these do, and then this kind of gives you just a general sense of how common they are. So 80% of all the RNAs in the cells are actually part of the ribosomes. So they're the RNA molecules that are part of the ribosomes. tRNAs make up an additional 15%, and then mRNAs make up 5%. So the actual message molecules, very little of the cell volume is used for the messenger RNA, and most of the RNA cell volume is actually used for the ribosomes and the parts of the cell that are necessary for producing protein. There are multiple components of the ribosome, and they're typically named by their size. So the 5S ribosome, the 16S ribosome, and the 23S ribosome. So these are all used by prokaryotic cells. And then eukaryotic cells tend to have a little bit different sizes. So through the process of evolution, the size of these rRNA molecules has changed. And they also share this small one, and they have an additional smaller rRNA molecule. So eukaryotic cells have 4 different versions. Prokaryotic cells have 3. And the nucleotide size is given here. So you can see that the Svedberg coefficient corresponds to the size of the rRNA molecules. Eukaryotic cells also use RNA for some additional purposes, and all of this stuff we'll cover in greater detail in later chapters. Telomerase RNA and RNA primers, so these are involved in DNA replication at the chromosome ends. So you need to have a specialized process in order to replicate the ends of chromosomes, which we'll cover next chapter, but RNA molecules are necessary for that. snRNAs, and kind of I would group all of these classes together. These are smaller RNAs, and these smaller RNAs are about, they can be maybe 20 base pairs in size. They can be a little bit larger, a little bit smaller, but they're around 20 base pairs in size, and these are typically regulatory. So they help process mRNAs, or they also help regulate the transcription of different genes, and they also can be used to regulate translation. So these are important regulatory molecules for controlling when and where different genes are expressed. So, why a different cell type is how a cell type is unique. They contribute to that process. RNA also serves as genetic material in some viruses. So most species in the kingdom of life use DNA, but there's actually viruses that use RNA and not DNA. So this is kind of an exception to the general rule, and the genome that's actually stored within the viral particles is RNA material. So an example of this is the tobacco mosaic virus, which was studied in 1956. And so for this particular virus, they actually store RNA, and that's what they inject into their host, is RNA. Coronaviruses, obviously you've heard about coronaviruses in the past year. Coronaviruses also have an RNA genome. Retroviruses are another class of viruses that use RNA as their template, and this RNA actually serves as a template. So in the host cell, the RNA is transcribed back into DNA. So this kind of reverses the flow of information from DNA to RNA, and RNA can actually revert back to DNA. And so this is using a DNA polymerase called reverse transcriptase. So these molecules we'll cover in more detail in later chapters, but these are very, very important from a biotechnological perspective, because they allow us, they've been hijacked by us in order to convert RNA into DNA. So there's certain reasons that you might want to take an RNA molecule that you isolate from a cell. Maybe you have an mRNA and you want to convert it back to DNA, and it's only because we've identified these enzymes that we have the power to do that.